Functions of membrane proteins. The lipids in the membrane of the plasma membrane of cells, they are very much similar of different cells. But the assortment of the proteins, that is very much different, and that has great influence on what the plasma membrane is doing and what the cell is doing. They are, for example, the channels. We are taking specific substances in or out through a hole. Most of these channels are selective. They are taking just one specific type of substance in. Therefore, many cells have different ones of these channels, selective channels, so that we can take different one for each one that's selective, in particular this one in. Then there are transporters. They are taking a substance and then they are shape-shifting and transporting it in, like a snake. They're taking it and then like a snake, they're bringing it in. For example, amino acids needed to synthesize proteins in the human body come through that way into a cell. Then there are receptors. There's a specific ligand called substance stuff, and that thing fits exactly therein. So having a specific shape or something, or somehow it works, it's not directly said how, that specifically this type of substance, this ligand, can dock onto it. And when it had docked there, it changes when the outside of a cell. It's not something differently because something else is now there. For example, antidiuretic things are docking in the kidneys that way on and changing their permeability. And then there are other things. There are then the cell identity markers, and there are glycolipids and glycoproteins, and for example, a major class of them are the major histocompatibility, and we are looking if a cell is of the same body as they are or of a different one, so we're checking for foreign ones. And when there are enzymes, for example, in the small intestine, there can be lactase in it, and if there comes now drunk for milk disaccharide lactose, they can take something in it, making a reaction and splitting it out again. So splitting up this disaccharide lactose in the small intestine. And whether which phase they are pointing these enzymes, where they are doing that thing, that job, that reaction. And then there are linkers, and they are taking a filament on the inside or outside, and that gives stability and structure to the cell, and also can help with movement, and they can also link with them to our cells. And that is according to Gerard Tatoga and Senegabowski's Principles of Anatomy and Physiology book on the pages 63 and 64, but this is no substitute for medical knowledge and also no medical advice. Hi, welcome. I'm trying to learn cytology today. Functions of membrane proteins. And after we've done it, we're gonna do arm strength training. And if you participate in the arm strength training, then it's at your own risk. Functions of membrane proteins. Generally, the types of lipids and cellular membranes vary only slightly from one membrane to another. The lipids in them, the phospholipids probably. So the lipids which are making the bilayer, I think I'm talking about, or all over something else. The lipids are almost the same to different membranes. No. The type of lipids in cellular, we're talking about of different cells having a similar product, similar thing for the membranes, whatever. Something of lipids are similar to others. Still, the membranes of different cells and various intercellular organelles feature remarkably different assortments of proteins, so lipids are similar in they are similar to other cells. The membranes are similar in different cells, but the membranes of different cells and various intercellular organelles feature remarkable different assortments. Does it mean how they are ordered? Assortments of proteins, which determine many of the functions that plasma membranes can perform. The types of the liquids are similar, but the assortments of the proteins is differently, and therefore the functions that the plasma membrane is doing is differently at different cells. Figure 33. Bunch of something. Shows several of the functions of plasma membrane proteins. Some transmembrane proteins are channels that have a pore or hole from which a specific substance such as a potassium ion, K+, can flow into or out of a cell. Most channels are selective. They allow only a single type of ion to pass through. Okay, there are channels, there are pores, they can go. What's the difference between a specific and a selective? So some have a pore or hole through which a specific substance, like potassium, can flow. Most channels are selective. They allow only a single type of ion to pass through. Okay, one, the pores, multiple, but also specific, and the other ones are selective, just one. Other membrane proteins act as transporters. Transporters bind a polar substance at one side of the membrane, and by changing their shape move it to the other side of the membrane where it is released. I did a few videos about these transporters. There was nothing said about such a simple explanation. So it basically just talks on. When it changes its shape, it pulls itself in, whatever. It changes its shapes, and therefore that thing is put in. Integral proteins, called receptors, serve as cellular recognition sites. These proteins recognize and bind a specific molecule, such as the... I think when they're talking about someone recognizes a specific molecule, they're just meaning it is able just to make chemical reactions with this one specific type of molecule. These proteins recognize and bind a specific molecule, such as the hormone insulin, or a nutrient such as glucose that is important for some cellular functions. So there are integral proteins, they are called receptors, and they bind to a specific molecule, like insulin or nutrient glucose. Okay. And I think that happens because they just fit together. Like two substances just never react together, they're just peacefully sitting there, but when other substances come together and suddenly it reacts. A specific molecule that binds to a receptor is called a ligand of a receptor. So it's the receptor and the ligand, they're binding together. Some integral and peripheral proteins are enzymes. Membrane glycoproteins and glycolipids often are cell identity markers. They may enable a cell to recognize other cells of the same kind during tissue formation, or to recognize and respond to potentially dangerous foreign cells. Some integral and peripheral proteins are enzymes. Membrane glycoproteins and glycolipids are called cell identity markers sometimes, because they may enable a cell to recognize other cells of the same kind. So they may enable a cell to recognize other cells of the same kind. So I think these glycoproteins, and I think I addressed in the last part only, these glycoproteins and glycolipids, they are binding to another cell. And as soon as they bind together with that one together, they are able to identify another cell of a cell that the glycoprotein has bound to. That is the way I understand it. Maybe I understand it wrong, but if it is, it's not that complicated. There is this hopeless lost cell, and it binds together with some detective, whatever, someone who can search for parts, whatever. Someone with an inspector together, and now we belong together. And that inspector, because they now are together, are able to find another cell which belongs to them. So it's just the lost child, and it's able to find the other lost children, which is not really much better, because it has someone with them which helps them to find some identity once again, if it still makes sense. Membrane glycoproteins and glycolipids often are cell identity markers. They may enable a cell to recognize other cells of the same kind during tissue formation or to recognize and respond to potentially dangerous foreign cells. I'm staying with what I have. It's a pretty clear explanation. If I'm wrong, I will pretty soon find out that it's wrong. The ABO blood type markers are one example of cell identity markers. Okay, there are some type of ABO blood type markers. There's some type of inspectors. The ABO inspector or detective. When you receive a blood transfusion, so a person can receive a blood transfusion, the blood type must be compatible with your own. So I mean the person who's getting the blood transfusion. What we mean is, I think, they are binding, these glycoproteins are binding onto the blood cells. And if it's a wrong type of blood cell, they are binding maybe on it, but when they're saying the detective is then not so much there to help, but to call more police to arrest that cell. Integral and peripheral proteins may serve as linkers. Okay, is that still the same? Some integral and peripheral proteins are enzymes. Membrane glycoproteins and glycolipids are often cell identity. Maybe some other way of the same we're talking about now. So some type of integral and peripheral proteins may serve as linkers, which anchor proteins in the plasma membrane, so the whole thing is the plasma membrane, of neighboring cells to one another or to filaments inside and outside the cells. They are linking between the other things. I don't know, here's nothing in particular explained, so I don't think it's that big of a thing for now. We're going to explain it in another part when it gets important. Wait for the biochemistry part. If you're here for biochemistry, you can go directly to the next part of biochemistry, and then we're going to do that. If you stay here and participate in the Armstrength training, that's very obvious. The leg sustain from the further part and breathe in and release the fingertips as fast as possible. And again down. And down. And breathe to the front. Up. And back. And if this exercise causes pain, then don't do it. Long arms and circle the fists. Back. And the other ear. Now the first exercise, arms out and press the fingertips together, not full force. And if there's pain at any moment at the elbows, the wrists, the fingers, or in particular the finger joints, then stop immediately. Have a little bend in the elbows and don't press with full force. And now the next exercise, press with the longer fingers. Again, a little bend in the elbow. And if there's at any moment pain at the elbows, the wrists, fingers, on particular finger joints, and stop immediately. And the next exercise. Press with the fingers into the fingernails of the other hand. If this causes damage at the skin or at the nails, then don't do it. Press. And give in. All only if it works with the angles of the wrists. Press again. And give. And again. Give. Again. Okay. Press. And then last time. Now the other way around, the left hand from the top. And give in. The other hand. And again. And now I think it was one more. think that was it all. So, one arm out and press on it and get in. Again. And now with square. Press your hands together. If it gives pain at the wrist, then don't. Press together, not full force. Over, up, over, down, in, and backwards. Over, over, down, in. And now we're downward. Press, middle, over, down, and again back. And now, I don't know what it's called. Very good. Arms to the side, circle in front. And backwards. And one arm in front. Shoulders back and down. Pull the arm in. Have flat fingers, relax the arm. Go with back of the shoulder to the palm of the sky. Pull the arm in. Shoulder down and back. Pull the arm. Have flat fingers, relax the arm. Go with back with the shoulder to the palm of the sky. And now go with moderate pressure over the middle part of the lower arm. And on the other side. And it was the Armstrength training. I thank you very much, and at the next time.